Hello, my name is Mina Chetinkara-Randell, and in this video I'm going to talk to you about workflows for reproducible data science. Or in other words, the results in Table 1 don't seem to correspond to those in Figure 2. So let's take a look at this random string of numbers. Well, they are a random string of numbers. In fact, they come from the uniform distribution. They've been randomly generated, but there's something special about them. We set a seed first, which means that the next person who runs the same line of code to generate 8 random numbers between 0 and 99 using the runif function in R will get the same set of numbers. So here's a very quick reproducibility tip for you. If your analysis involves generating random numbers, make sure to set a seed so the next person who runs your code can get the same results as well, or the same values as well. Let's look at a few more numbers. The number 70. There was a study in 2016 where 1,500 scientists were surveyed, and more than 70% said they have tried and failed to reproduce another scientist's experiments. That's a striking number, but also a high bar to reproduce somebody else's work. What about the number 50? More than 50% said they have tried and failed to reproduce their own experiments, and that is a very striking number. The number 965, that's how many Google Scholar yields there are just in the year 2021 when you look for the search term reproducibility crisis. Now, obviously, reproducibility crisis is a bit sensational, even though there is a reproducibility crisis. And it tends to be that if you have, a sensational title, maybe your paper is more likely to be published. There are some studies around that. So this could potentially be a biased result, the fact that this is a high number, but regardless, that's a high number for just 1 year. 1992 is the earliest reference that I was able to find to reproducibility research. And here is what that paper is. It's called Electronic Documents Give Reproducible Research a New Meaning. And I want to show you a few excerpts from that. So the author said, in 1990, we set this sequence of goals. Learn how to merge a publication with its underlying computational analysis. Teach researchers how to prepare a document in a form where they themselves can reproduce their own research results a year or more later by pressing a single button. Learn how to leave finished work in a condition where coworkers can reproduce the calculation, including the final illustration by pressing a button in its caption. Prepare a complete copy of our local software environment so that graduating students can take their work away with them to other sites, press a button, and reproduce their Stanford work. This is where the study was published. Merge electronic documents written by multiple authors, and export electronic documents to numerous other sites so they can readily reproduce a substantial portion of our Stanford research. I would say that we haven't actually achieved all of these goals entirely yet, and that's striking. So here's another striking excerpt from the same paper. It says, now that we've begun using CD-ROM publication, such technology for back in the day, we can go much further. Every figure caption contains a push button that jumps to the appropriate science directory or folder and initiates a figure rebuild command and then displays the figure, possibly as a movie or interactive program. This is the sort of thing that would be fantastic to see that is still not commonplace in journals. In fact, there was an article published in 2018. This was the first, I believe, published document that had live code associated with each of the figures. So you can see one of the figures there, and there's a little blue button that says R script. The idea is that you can click on that and you can actually reproduce that figure. So what was said in that 1992 paper became a reality in 2018, and it is still not commonplace in scientific publication. Even though there's more move towards reproducibility, even some journals requiring that code be submitted and data be submitted along with the publication, we still don't have this push button reproducibility thing for, all of the analyses in a paper. It is difficult to achieve for a variety of reasons. So I want to, before we go into talking about workflows for reproducibility, I want to set the stage a little bit, just so it is clear that, you know, we've talked about some ambitious goals, and the goal in this short video isn't to necessarily give you workflow tools to achieve all of them, because that would be, you know, years and years worth of work, not just a short video. So when we talk about reproducibility, really two words come to mind: replicability and reproducibility. So generally, I defined these two as they are both, when we're talking about replicability and reproducibility, we're trying to answer the same research question. We want to get to same results. But in the case of replicability, we're talking about a new data set. Say you have conducted an experiment on a subpopulation, and now you're doing so in another subpopulation. Versus when we talk about reproducibility, or particularly computational reproducibility, we're talking about the same data as well, where you might be thinking, how would we not, you know, get the same results if we're asking the same research question and working with the same data? Well, turns out that's still not that simple. So that's the reproducibility definition that we are going to be working with, not the replicability definition. If you are more interested in replicability, I do recommend at a minimum the ASA statement on p-values. That I think is a good read in terms of some of the things that might go wrong when it comes to thinking about replicability. So here's a quote from Mark Holder that says, "Your closest collaborators used 6 months ago, but you don't reply to emails." This is to say that when we're talking about reproducibility, we're not always talking about reproducing other people's work. Sometimes we are talking about reproducing our own work and how difficult that can be. So, for example, let's take a look at this very simple table where we have regression output for predicting, you know, an attribute of, you know, a set of irises. This comes from the iris data, their petal length from their sepal width. Okay. And here is the regression output. And then further down in the paper, we have a figure that shows the relationship between these two variables. Look carefully at the regression output and the figure that we have provided. There's a mismatch. The regression coefficient for the slope is negative, but in the figure it's positive. And this is not, you know, this is the sort of mistake that can happen pretty easily. How? Well, you might start writing some report, and then you do your analysis, you fit a model to predict one variable from the other, and then you say, all right, let's print the model summary. I'm going to copy and paste the output and put it into my report. Then I do a bunch more writing, maybe I'm pages ahead. Then I say, let's visualize the relationship as well and put a figure here, and let me copy and paste the figure. And well, when I do that, what I'm seeing is that there's clearly two groups of flowers that are in this data set. And maybe you decide, you know what, this analysis doesn't make sense like this. I think I may want to analyze the setosas separately and keep the other two in my data set. So how about I write maybe some more narrative about why I omit that one group and then fit another smooth curve to, or a line to, the remaining data. And now this looks more like something you might want to include in your paper, and you go on your merry way. Well, what happened is that now you have this mismatch. And again, imagine there's pages of other writing in between these. Obviously, if you're being quite careful, you would go back and fix your table one as well. But now you as the human need to remember to do that. Or maybe one of your collaborators made the other fix, and you were the person responsible for a particular section, and somehow or other an oversight occurred, and this is what you're left with as your final manuscript. So that's the sort of thing that we are trying to avoid. So how do we make our research reproducible? Well, here's a quote from David Donahoe about, and who's paraphrasing kind of John Claerbout, who was the author of the earlier 1990s paper. It says, "An article about computational science in a scientific publication is not the scholarship itself. It is merely advertising of the scholarship. The actual scholarship is the complete software development environment and the complete set of instructions which generated the figures." This is important to keep in mind because oftentimes we think about the scientific publication as the end-all be-all, because that's the thing that gets credit. That's the thing that we get credit for. But the work that gets poured into creating that is really where the science is happening, not the published article itself. So what we want to do in order to be able to achieve that is that we want to make our raw data available and accessible. We want to make our code and documentation to reproduce the available analysis available and accessible, and we want to make specifications of your computational environment available and accessible as well. Without these three components, it's impossible to reproduce one's work. You might make your data available, but if your code isn't available, chances are it's going to be very difficult for someone to reproduce your work, unless if your work was so straightforward. But really, no, published research is so straightforward. And similarly, even if you have the raw data and the code and the documentation available, running your code in a different computational environment could potentially result in different results due to different software versions or something like that. So really, we need all three of these puzzle pieces in place. And the most important tool is the mindset when starting that the end product will be reproducible, says Keith Baggerly. This means that you really want to start with this reproducibility mindset to begin with, not get to a bunch of results, publish about them, and then at the time of maybe submission, think, I wonder which of all of the things I have done I should include as the code that goes along with this paper. Because that is going to be tedious and not feel very rewarding, and chances are will just feel tedious, and probably you won't be able to go back and easily find all of the bits and pieces you need to reproduce your own work even. So if this is kind of the reproducibility kind of horizon, where on one end you have nobody, not even yourself, can create any part of your analysis, and at the very other end you have push button reproducibility in published work, like that article we showed earlier. You want to target that ultimate goal of push button reproducibility, but I think you really want to try to be happy in a zone that gets close to that as well. And if you're earlier in this kind of horizon, just try to take one step forward towards that target every time with each new project. There is no one-size-fits-all solution for computational reproducibility. So I am going to make some suggestions here, and chances are some of them will apply to your work and some of them will not. And similarly, if you read about reproducibility elsewhere, you'll come across some solutions that might be very applicable for your case, and some where you feel like this does not at all work for me. For example, I talked about making your data publicly available. Some of us work with data where that's simply not going to be the case. It simply has to remain private due to privacy concerns or personally identifiable information. So there are still reproducibility approaches for working with data like that, but a suggestion of just post it on the web is clearly not going to be a solution that works for you. But even though there is no one size fits all, the following 8 principles that I'll go through might help. The first one is you want to organize your project. So Jenny Bryan says file organization and naming are powerful weapons against chaos. So if we're on this kind of level of organization spectrum, and on one end this is what your desktop looks like, and on the other hand you have a very organized kind of boxes and boxes kind of situation, again, you kind of want to be getting as close to this final goal as possible. How do we do that? Let's say you have a simple analysis where it lives in a folder, and that's important with each new project. Create a new folder, and in it maybe you have something like a raw data folder, a process data folder, and a manuscript folder, where your manuscript is an R Markdown document, which is a computational document that houses both your code and your narrative. In a more complex analysis, maybe you again have the raw data and the process data folders. important to keep these distinguished. So you have your raw data, which is how you get your data, and that's holy. You don't want to touch that. But obviously, you're going to want to process that data a bit and probably save an interim version of that. So instead of overwriting your raw data, you want to save that separately, and that's probably the data that you're using for your analysis. Now, if your analysis is more complex, you probably won't want to put all of your code to create that analysis into a single R Markdown document that has to, go through a lengthy time to run all of your code each time you knit it. So what you might do is you might have some scripts that are run in a particular order that generate some figures that ultimately get sucked into your manuscript. It's important to stick with the conventions of your peers. So if this is kind of the folder structure that your collaborators, your lab uses, go with that. If you use something slightly different, which different types of data might require slightly different organization, then stick with that. Recommended reading for you is a blog post by Lucy D'Agostino McGowan that she wrote at the time she was finishing her dissertation, where she compiled resources that she used for completing her dissertation that include some reproducibility tips as well. Write READMEs liberally. I would say every folder needs one of these things. Don't assume you're going to remember what's in that folder. So if this is what your project folder looks like, where in a raw data folder, let's say you have a bunch of data sets, some CSV files. These are just ones I grabbed from the NYC Flights 13 data set. So these are the data sets that come in that package. But I have a README that says, this folder contains the raw data for the project. This is where I downloaded all the data from on this date. And then I say, here is what each one of these files are about. Don't assume that you're going to remember this information. Keep your data clean, tidy, and machine readable. What do I mean by that? This is the sort of data I encounter a lot, as I am I teach lots of courses, so I end up with lots of kind of student rosters. So let's say in this example, I have some students and I have their exam grades. You can see that I have this like merged column header for exam grade, and I am giving 2 exams in my class. And I also have recorded the major that these students are majoring in, but I'm missing some data for at least one of them. When I look at the exam grades, a person missed their exam, another person was sick. I've also used color, of all things, to identify low participation, which, very well might be true. If you have a spreadsheet in front of you with your students' names and you want to quickly look at, oh, I should watch out for these students, color can be a very good way for a human to quickly get sense of their data. But it is not machine readable. So instead, you would want to record code and document non-code steps to go from one version of this file that's not machine readable to the other version that's on the right-hand side that is machine readable. And also, whenever you're doing this sort of conversion, write a few tests for yourself. Whatever the number of students you had should be the same number of students on the second data set as well, for example. You can see that I don't have any sort of merged headers. Color isn't being used to identify participation level. Students with low participation actually have a column dedicated to identify this. So this data set on the right-hand side can now be easily turned into something like a CSV that can be read in and parsed programmatically as well. Thinking about kind of naming structures and whatnot, I recommend this paper in genome biology called Gene Name Errors Are Widespread in the Scientific Literature. In fact, I believe there has even been a shift to change how genes are named so that Microsoft Excel does not transform them to dates, but this used to be a common issue because of the naming structure of some of the genes that led, as you can imagine, to lots of reproducibility issues from research that where data was opened in Excel documents and then some of the gene names were mangled because they were converted to dates. Another recommended reading is data organization in spreadsheets. And so in this paper, there's some really good advice about how to organize data in spreadsheets. If you are a spreadsheet person to begin with, I think there's still bits and pieces that you'll learn from this paper, and some of it might make you think, how is this not obvious to other people? But it's not always the case, especially for researchers where this is not necessarily the medium where they tend to record their data to begin with. So this is a great reading for somebody working with data. It is also a great reading to share with collaborators who are more on the end of data collection. Number 4 is comment your code, and comment it liberally. So here is an example of a code comment. We are doing, we have a scatter plot here, and we are doing, fitting a smooth curve to the points using lowest smoothing. And when you do this, one of the things you specify is a particular span, right? So here I have specified 0.375, and I have commented my code on top. I have a comment that says use lowest smoothing. But the thing is, what is 0.375? Did I stumble upon that number? Did I try a few things and that's the one I landed on? Is this some prior I have based on other research on the same topic? My comment doesn't say anything about it. So when we say comment your code, it is useful to comment the code to say what you're doing, because sometimes it may not be so easy to remember even what you were doing when you were writing the code if you come back to it, a couple months later. But also comment it to say why you have done the thing you've done. Number 5, use literate programming. This is probably one of the cheapest, in a way, ways of being reproducible. So very nice literate programming system is the R Markdown ecosystem. So here we have what we call a computational document that is written in plain text. And you can see that in addition to prose, that is, say, on line 21 and 22, and also 29 onwards, we have our code chunks that are, say, starting in line 14 and starting in line 24. And these are marked by these kind of fences that are marked by the backticks, and anything in there is our code. So what happens when we knit this document is we get a nicely rendered file that has both our R code but also the results as well. So you can see, for example, under the model heading, we are able to see the output of our model without doing additional work like copying and pasting. Similarly, if you take a look at what is happening on the source side, on the left-hand side on line 29, where we have, some inline R code that tells us how many unique species there are. And then you look at the output right about in the middle of it, it says the original data set has 3 species, but we will only work with 2 of them. Those numbers 3 and 2 are not handwritten, but are being calculated with the inline code. So our Markdown allows us to have code chunks that do a computation, and then we'll print out the result. It also allows us to have inline code that helps with our narrative as well. And if this is going to a venue where the audience does not need to see the code, you can change code chunk options to hide those without actually removing the code from your document. So here I have set echo equals false to say don't show the code. And so now when I knit it, the code is still there. The document is still reproducible. It's just hidden from the final output. So, which means you can imagine writing an entire scientific article doing this, and in fact, lots and lots of researchers do so. If you are interested in learning more about how to use R Markdown, I recommend the book R Markdown, The Definitive Guide, and also the R Markdown Cookbook. And this second book particularly helpful if you have already been using R Markdown and are able to do kind of more of the simpler things of put a simple analysis in an R Markdown document, but you're running into issues in terms of runtime or in terms of how to organize a project around our Markdown, or how to do some, advanced styling things, and the cookbook can be quite helpful for that. Number 6, use version control. So this really is easier said than done in a way, because learning version control, it comes with its own learning curve, and I would say a steeper learning curve than R Markdown, but not as scary as it sounds also if you are using the right tooling. So here, for example, I have this R Markdown document, and what basically I want to do is I want to keep that R Markdown document in a project where the changes are tracked by Git, and then I can host that on GitHub as well. And the hosting side is helpful both for public sharing, or you could host things on a private repository, even if just for yourself, but also to be alternatively to be shared with collaborators as well. So the goal is really to minimize any situation where you're attaching an analysis to an email and sending to somebody, but instead you're tracking your changes with Git and then hosting them on a GitHub repository where the appropriate permissions are given to the appropriate people, whether that means publicly available to the rest of the world to see or just for your collaborators. I would recommend, if you are going to be kind of learning to use version control with R, instead of reading about Git kind of generally as a version control system, for all sorts of software projects, I would recommend this research called Happy Git and GitHub for the User, which is basically for using Git along with R, and particularly using the RStudio IDE, but also their approaches. They're highlighted if you're not RStudio IDE user. But a really nice starting point, I think, if you're working in R, is to use that Git pane and the RStudio IDE, so you can really visually see what your changes are. And with the push of a few buttons and clicks, you are able to commit and push your changes to a repository. So this would be a resource I strongly recommend for learning to use Git along with R. Number 7 is automate your process. As much as possible, try to organize your work where it is automatable. So for example, if you have some scripts that need to be run in a particular order, don't try to commit that to memory. Actually name them in a way that it makes it clear the order in which they're going to need to be run. And then maybe a really cheap way of doing that is to add another script to the very bottom of that chain that says analyze, and that simply sources each of the other files in a particular order. So this is a very kind of a quick and dirty way to say run the following scripts in a particular order. Now, what this lacks though is watching for any sort of changes in the code and whatnot. So this is basically saying run everything regardless of whether I've changed anything in step 2 or step 3. A more kind of principled but a more complicated or complex to learn approach would be to use something like the make tool, which is going to do basically what we outlined in the previous slide, except using a make file. And here the make files are a bit smarter about kind of have changes occurred and really which bits of the analysis need to be rerun. R-based approach to this sort of to the make approach, it uses this package called targets. It is a new-ish package, I would say, developed in the last couple of years, but there was a precursor package to this as well. So this is not kind of the first approach to solving this problem within R. And it has a really extensive manual. And the nice thing about these sorts of approaches, either with Make or using the targets package, is that they are extensible to a lot more complex projects than, say, what you might want to organize within a single or markdown document. And the idea here with targets is that you write these reproducible pipelines for doing your analysis, and targets learns how your pipeline kind of fits together and skips costly runtime for tasks that are already up to date and runs only the necessary computation. And finally, share your computing environment. And this is another one of those easily said than done things as well, and probably the one that requires kind of the most technical infrastructure of all the other tips that I've mentioned. A very kind of, again, a quick and dirty way of sharing your computing environment is to use something like RStudio Cloud or any sort of RStudio server where you have control over how that server was set up, and the next person who is trying to reproduce your work is able to find themselves in that same setup, whether via the sharing of an RStudio Cloud project or by being able to log into the same server where you are doing your computation. So this is what an RStudio Cloud project looks like. It looks just like the RStudio ID, just in your browser. Another approach is using Binder, which is something you can tag on to your GitHub repository, where it will basically, within your GitHub repository, will launch a computing environment that has the necessary software and the versions of packages and whatnot already built in, so that code that's living in that repository can be run in this environment set up by Binder. And finally, a third approach would be using Docker. So here you are creating a container that basically has the whatever software environment you define with whatever versions of your software and the packages that you might be working with, and it basically has a specification in mind, and the goal is that somebody else can use your Docker container, the container setup that you have prepared, to be able to run your code kind of within the same environment. So we've talked about RStudio Cloud, we've talked about Binder, we've talked about Docker. A few recommended readings, especially for talking about Docker. There's a nice write-up called An Introduction to Docker for Reproducible Research, and also another one that is Packaging Data Analytical Work Reproducibly Using R and Friends. And each of these basically look at approaches for kind of replicating the computational environment where the analysis is happening and being able to share that with others so that your analysis can be reproduced by them. So the 8 tips that we talked about were: organize your project, write readmes liberally, keep data tidy and machine readable, comment your code, use literate programming, use version control, automate your process, and share computing environment. All of this at once can seem like a lot of work. So if you're thinking, oh, this is going to be a lot of work to be reproducible, my recommendation for you is find where you're at in this spectrum, and for your next project, that just try to take one more step forward. And if you're at step 0, simply start by using a particular organization for your project. As I said, if there's an ecosystem you're already plugging into, use their standards, how they tend to organize their projects. And if not, try a few things until a particular approach starts to feel good for you. But take one step in kind of the reproducibility spectrum at a time, as opposed to trying to do everything at once. I would also recommend this article called Good Enough Practices in Scientific Computing. That is like another one of these 10-step type articles. And what I like about this article, that it's not just best practices, it is good enough practices, in the sense that it's not always possible to be able to apply the best. But if you can be simply good enough, or at least better than where you're at starting with, that is an important step towards reproducibility. Some of the painful bits can be things like coming up with good names for things, thinking about how to structure various stages of data cleaning, going back and redoing stuff and how to document that, adding interim steps once you have, say, a workflow going. Maybe you have numbered your scripts and you have kind of been working on them in that same order, and then you all of a sudden realize there needs to be a step, 4B between your step 4 and step 5. Keeping track of the order of things, and also the clutter of unneeded old stuff. Sometimes you try certain things and you know that you're not going to use them. Say you have created a figure and you're like, I don't think this is going in my paper, but I kind of don't feel like deleting this code right now either. So these can be, nuisances to reproducibility, things that clutter our project space and try to make us and make it difficult for us to feel organized. And there aren't great solutions necessarily to all of these, but it's important to know, one, that these painful bits exist, and number two, trying to come up with a workflow that works for you for working through some of these painful bits. I'll tell you my approach is, whenever I have I start collecting some scripts maybe within a project that I'm currently not using, I will put them in a folder called unused and keep that there until maybe that paper goes to publication, or I'm done with that project or that talk, because I don't want to get rid of it, but I also don't want it cluttering my workspace either. Looking into the future, we talked about this quote, the most important tool is the mindset when starting that the end product will be reproducible. Where when we're thinking about mindset, I think another way of thinking about this is that the second most important tool is training. If you want to have this mindset, you need to be trained to be reproducible. So if you already have your own workflows and some of what I said feels orthogonal to your own workflow, that's probably not feeling great right now. But what if from the very beginning of your data analysis days, you actually had a reproducible workflow to begin with, where you wouldn't be sitting through this talk perhaps at this point? For me, I feel like part of my role is to convince data scientists to adopt a reproducible data analysis workflow, but then also when working with new data scientists who don't have any other workflow, we want to train them to be reproducible. And as an educator, I feel like my piece of the pie is to instill best practices in students for reproducibility before they set out to do research. And chances are you also, in addition to being a researcher, are going to be involved with educating the new generation at some point. So you want to think about your own workflows and the workflows that are recommended for reproducibility, and always thinking about how can I also teach this to students, even if they are at the very beginning of their learning to work with data journey. So I'll give you an example of how we do this in my teaching. So whenever students are doing computing, they don't work with locally installed versions of their software. They just go to their browser and they log in, and they maybe go to something like RStudio Cloud or another RStudio server, and they have an Markdown document that they're working with, generally one that I have scaffolded for them, and they knit that document. And once they knit that document, they can see both the code and the output and the pros all in one place. Each of these documents live in projects that are Git tracked, and each of them are a Git repository. So when students are working on an analysis and submitting an assignment, they're submitting that as a GitHub repository. This means that they are learning to use tools for version control as they're learning to do data analysis. And at some point when they venture on to doing research, this tooling is, already second nature to them. So thank you very much for listening so far. If you are interested in the slides, particularly for getting some of the links to the recommended readings and the tools that I had, you can find these slides at bit.ly/repro-ds-21. Once again, my name is Mina Chetinkara Rendell, and you can find me on Twitter at Mina Bojek, or on GitHub at Mina Chetinkara Rendell, where I tend to house a lot of my projects and version control them and share them with the rest of the world. Thank you and bye.